Hello everyone, welcome to today's session. If you are new to our channel, please subscribe it for the daily videos. The mechanism of neurotransmission. Neurons communicate with their target tissues at synapses into which they released chemical substances called neurotransmitters or ligands. As this communication is mediated with chemical substances, the process is called chemical neurotransmission and happens within chemical synapses. Types of neurons and synapse structure. Each synapse consists of the presynaptic membrane, postsynaptic membrane, and synaptic cleft. Inside the terminal button of the presynaptic nerve fiber, numerous vesicles that contain neurotransmitters are produced and stored. When the presynaptic membrane is depolarized by an action potential, calcium voltage gated channels open that is found in the membranes of the terminal buttons. This leads to an influx of calcium ions into the terminal button, which changes the state of certain membrane proteins in the presynaptic membrane and result in exocytosis of neurotransmitters from the terminal button into the synaptic cleft. After crossing the synaptic cleft, the neurotransmitters bind to their receptors on the postsynaptic membrane. Once the neurotransmitter binds to its receptor, the ligand-gated channel of the postsynaptic membrane either open or close. These ligand-gated channels are ion channels, and their opening or closing alters the permeability of the postsynaptic membrane to calcium, sodium, potassium, and chloride ions. This leads to a stimulatory or inhibitory response. If a neurotransmitter stimulates the target cell to an action, then it is an excitatory neurotransmitter acting in an excitatory synapse. On the other hand, if it inhibits the target cell, it is an inhibitory neurotransmitter acting in an inhibitory synapse. So, the type of the synapse and the response of the target tissue depends on the type of neurotransmitter. The neurotransmitter released into the synaptic cleft acts for a very short duration, only minutes or even seconds. It is either destroyed by enzymes such as acetylcholinesterase or is reabsorbed into the terminal button of the presynaptic neuron by reuptake mechanism and then recycled. The best known neurotransmitters responsible for such fast but short-lived excitatory action are acetylcholine, norepinephrine, and epinephrine, while GABA is the major inhibitory neurotransmitter. Repeated synaptic activities can have long-lasting effect on the receptor neuron, including structural changes such as the formation of new synapses, alterations in the dendritic tree, or growth of axons. An example of this is the learning process. The more you study and repeat, the more synapses are created in your brain and enable you to retrieve that information when needed.